Alright, let's go to embryology next. After the sperm goes through the uterus, the fallopian tube, and about 300 make it. They get to the ovum. And then when we talked about the sperm, we got the tail here, of course, that drives it forward. We got the neck, and then we got the DNA stuff, the cell itself, the genetic material, and then on top we have that cap. And the cap is an acrosome. And the acrosomal enzymes, as the sperm wants to go through, disperse the cells of the corona radiata. And then the spermatozoa reaches down here at the blue area. That's the zona pellucida. And it reaches the zona pellucida, and the acrosomal membrane dissolves, and enzymes facilitate the passage into the ovum. And then once that one is in, we have an action potential that goes through the membrane, and it's called a cortical reaction, and that leads vesicles to go and get released, the cortical granules. They create a wall for other sperms to come in, so they become a polyspermy block, preventing the entry of other sperms. So once that one is in, its head swells up and it becomes a male pronucleus, and the ovum completes its second maturation process division to form the female pronucleus. And then we get a fusion of the two, and that creates a diploid zygote. That's the word. A diploid is the 46 chromosomes. You have a copy of 23. They both were haploids. They only had 23 chromosomes, but now combined they have the 46. And from that process on, we start then multiplying by mitosis after about 30 hours. The male pronucleus is the one that determines the sex, as it carries an X or a Y sex chromosome. The female has an X always, and so it will create a boy if it's an X and a Y, or a girl if it's two X's. Now that the egg is fertilized, it takes about 4 to 5 days to migrate through the fallopian tube into the uterus. During this period, it multiplies a lot of times, and first it becomes a morula right here, and once it enters the uterus, what happens is the zona pellucida regresses and lets it implant in the uterus. Basically, it prevents the implantation. Zona pellucida prevents implantation in the fallopian tube, but once in the uterus, we want an implant. We want what they call a nidation. We want to do that, and the zona pellucida is gone, and then it becomes a blastocyst. And the blastocyst has an outer cellular envelope, and that's known as a trophoblast, and it has an inner cell mass, and that's the embryoblast. And now we can embed that in the endometrium of the uterus, and that process is known as nidation. That brings us to the placenta. Upon nidation, the trophoblast, so the outer structure here, the trophoblast, forms villi that become the fetal blood vessels. So that goes like right in here then. They form a discoid placenta. The placenta is going to be right up here. And they do that together with the uterine mucosa. So this is where the new ovum, the fertilized ovum, reaches and has a connection with the placenta. And then, with the uterine mucosa, as in the placenta. The placenta nourishes the growing embryo through the umbilical cord, taking over gas and nutrient exchange between the maternal and the fetal bloodstream. It also ensures the stability of the pregnancy by secreting placental hormones. So that's another thing the placenta does. It's really a gland. It secretes estrogen, progesterone, chorionic gonadotropins as well. Looking at it, the placenta is shaped like a pot with about a diameter of 18 centimeters. It weighs 450 to 500 grams. The bottom is formed by the uterine mucosa, forming intervillous spaces that are filled with maternal blood that exchange about 3 to 4 times a minute. So that's right here. And that's the interstitial blood. And then we have the lid, which is the top here, chorionic plate, that is formed by fetal trophoblast. So that's that outside cell structure of the blastocyst. We have 15 to 20 structures that branch like trees that are formed by villi reaching into the maternal blood. So these are these things, they go into the maternal blood. And there I have this weird name, cotyledons. I don't know, gotta be French or something. The placental barrier. The villi are lined by an epithelium and carry the fetal blood vessel. So those are these villi here that go in, and they have fetal blood vessels inside of them. And the gas substance and substances are transported through the epithelium and the connective tissue so that the maternal and the fetal blood do not mix. So there's a barrier, the placental blood barrier. And then the umbilical cord is a gelatinous connective tissue envelope that carries an umbilical vein, which brings oxygenated blood into the fetus, and two umbilical arteries, which bring deoxygenated blood away. Veins lead into, arteries away. In this respect here, we're talking about the fetus. Away from the fetus or to the fetus. And then after we give birth to the baby, we also have to give an afterbirth, and that's the birth of the placenta itself. And we have to examine that very well, because that can lead to problems if there is ruptures and these structures are not fully brought out of the uterus. And so we have to really look at that, because we don't want bleeding, because that could be a big potential problem for the mom. So we examine that, and we show the elevated areas, and those are these trophoblast cotyledons, though reaching into the maternal blood, and then they are covered by a thin membrane, and that's the disputa basalis. And when that's all good, then we're good. Okay, let's talk about the embryonic development of. We have an embryoblast. That's this baby right here, right in here in this middle part here. And we have a two-layered embryonic disc. We have a endoderm right here in the yellow part, and we have a ectoderm here in the bluish part, the outer part. In the middle one, in the inside, we actually have another layer that's created. That's the mesoderm. We have those two plates, the outer plates, the endoderm on one side and the ecto on the other side. They're covered. The endoderm is covered with a yolk sac, and the outer one, the ecto, is an amniotic sac. The yolk sac regresses slowly, while the amniotic cavity grows to about 1 liter of amniotic fluid, which then nourishes and protects the fetus. At about, and now you see this this embryonic disc here, this piece here. We see it right here, right there. At about 16 days, we have a narrow groove, and this here, the narrow groove, the primitive streak. On the ectoderm side, it deepens and it folds over itself, and it creates an inside cord. And that's the nodal cord, chorda dorsalis. That becomes the spinal cord area, the spinal canal, basically. And that's underneath the ectoderm. And then cells in this area also migrate inwards and form the mesoderm. And what I like here about this picture here is you see the ectoderm, the mesoderm, and the endoderm, these three primitive cell layers, and what they become. The endoderm becomes the viscera, the mesoderm muscle bones, and the ectoderm skin and nervous system. And then we move forward, and at first, like a shield, the embryo constricts at both ends and forms a basic trunk at about 4 weeks. And then the head grows rapidly and takes up about 1/3 of the length. It's shaped and is created by the primary and ocular vesicles. So that becomes a big structure. At about the 5th week, the upper and the lower limb buds appear on the lateral wall. And you can see that little bit here coming out here. These are, of course, the buds are down in here. The trunk bulges due to the cardiac tube and the liver that is growing. So it goes forward, bulges forward. During the 2nd month, the embryo bends markedly at the head, and we have a cervical cephalic flexures that get created, and the head now takes up about half of the body We can see eyelids, we can see nose, we can see lips, we can see the pinna, the ear, as well as the chin. And then fingers and toes are also seen as little rays that come off the bud limbs, the limb bones. From the beginning of the 3rd month, the now we call it now a fetus, and the organ systems differentiate. Growth happens now. Growth happens in spurs relatively small up to 26 weeks, and then it accelerates it thereafter maximally. So the first the systems get developed, and then we can add size to them. The fetal is grown by ultrasound, so we can monitor the fetal growth. When we measure the fetus, we measure the distance between the parietal bones, and that's the biparietal diameter, times 5.5 gives us the body length in centimeters. So that's pretty interesting. At full term, the crown to heel length of a fetus is about 49 to 51 centimeters. The crown to rump, which is the sitting length, is about 33 centimeters. An average weight is about 3.2 to 3.4 kilograms. Below 2.5 kilograms is known as low body weight. When we get a fetus, we need to look if the fetus is mature. We have some signs for that. Signs of fetal maturity are lanugo hair. It's a fine little hair that covers the body. Fingernails project beyond the digits. The testes has reached the scrotum, and the labia majora just covers the labia minora, the ladies, the girls. Other criteria is the skin color, turgidity, that's the hydration, as well as the subcutaneous fat. We want to see stiffness of the nasal and the auricular cartilages by the nose and the ears, the presence of some reflexes, the respiratory state, heart rate, as well as muscle tone. The epiphyseal centers, the growth of the bones, those centers where they grow, can be seen radiographically if wanted. That's also a sign for fetal maturity.